Hello everyone. Today I plan to give you an overview of the research I have conducted so far towards my portfolio. I appreciate any constructive feedback to assist me. Okay, so firstly, to expand my epistemological understanding, I have turned to Derive in 2012, and I read his article and it expanded my epistemological knowledge of biology to generalize it as the life science. Biology is the study of life from an anti-vitalist approach, which I have now learned is when phenomena is broken down into its basic components. Yet in doing so, we as living organisms are experiencing life and simultaneously attempting to break it up into purely logical segments. This process in itself did not develop from pure logic, but from history, yet epistemological approaches are rarely applied in biology. In the breakup of life into its separate components allows for a more analytical view of our biological world. An example of this would be that a plant is a living organism, yet photosynthesis, a broken up component that makes up that plant's ability to live, is in itself not seen as life or living. The scientific method is the largely accepted method in which scientific data is produced. Using the diagram as a prompt, the scientific method poses a question, researches the already available information, then poses a hypothesis, which is a proposed explanation to your question, which includes the variables that you would like to test. Then you test the hypothesis, analyze the data, and then from your analyzed data, you make a conclusion. Either your hypothesis was wrong, so your explanation was wrong, so you make a new hypothesis, a new explanation, and retest. Otherwise, if you were correct, you retest anyway to double check yourself. You communicate your findings and then propose it to the scientific community. The scientific community will have a look at it and either agree with your hypothesis or disagree based on your methodology and your data. From an epistemological point of view, the scientific method in itself is a tautological structure, meaning that the knowledge is built by repeating itself with different terminology and contradicting the previous knowledge if necessary. For information to be considered substantiated in science, it needs quantitative data to deem the hypothesis true or false. After my original meeting with Brian, I decided to base my pedagogy around course relevance. After researching relevance as a pedagogy, I found there are different types of relevance, even the teacher's mannerisms and persona in the classroom and how the students can relate to that. After researching the method, I honed in specifically on outside course relevance. This method is choosing parallels between the content and events, interests, or desires that lay outside the classroom. Using this pedagogy has resulted in an increase of student engagement with the content and a greater motivation for students to learn. With the inquiry plans in mind, I decided I also needed to find a method to structure my lessons. To do this, I have turned to the 5E framework. The relevance topic will be used in the engage stage of the framework, and the 5E model is based on a constructivist design to increase the metacognitive process of the students while they're in the classroom. Okay, so now we're on to my content knowledge. So I have chosen the topic of genetics for my portfolio as I felt this was my weakest link. I have decided to specifically use the mutations topic within the genetics unit. So firstly, I looked at any terminology that I didn't understand and then turned to my textbook that I had left over from my bachelor's to clarify anything. And there was a lot that I needed to clarify. So firstly, mutations that occur within DNA replication, there is two types, point and frameshift mutation. For the point mutations, there is two subcategories. So you've got the missense mutation, which is swapping out one base for another base, causing the mutation. So you're swapping out, in this particular example, this is the normal mRNA strand that's to be copied into your amino acids to create your protein. And this one has swapped out a guanine with a cytosine. And the second type is a nonsense mutation, which swaps out this one here for a stop codon, which truncates the protein shorter than the length it was supposed to be. Now we've got the frameshift mutation. So that is exactly how it sounds. So this is your normal mRNA. So it's like this strand here, and the ribosome goes along and reads it. And rather than starting from your start frame to translate the code into its amino acids, the frame has shifted across and now it's starting one base over. So as the code translates, every translation is now translating to an incorrect amino acid from what the mRNA originally was coding for, which is a lot more harmful than your point mutations. Okay, now we've got cell division and non-disjunction. So I found out that non-disjunction is essentially the unequal dividing of the chromatids during cell division. So this is your normal cell division, and as everyone will be able to do your basic maths, everything is divvied up evenly, and then when you get to your gametes, every gamete has two chromatids in there. Now, when you get to your non-disjunction, so here's two separate examples of that, and essentially it's just being divvied up unevenly. This cell here has 6 chromatids, this one only has 2. So then when you get to your gametes, these two both have 3, these only have 1. And because the gametes are your sex cells, this can have effects within the offspring that come of that. Next, we've got damage by mutagens. So mutagens is anything that reacts with the DNA directly and creates a mutation. So this can range from chemicals to the sun UV radiation to ionizing radiation, X-rays, anything like that. And all of those three make up the base of this entire unit. Now that we have the basics, we need to dive a bit deeper and understand that non-disjunction leads to aneuploidy. Aneuploidy, I had no idea about, so doing some research, ploidy is the number of chromosomes in the cell, which for a human is 23 pairs of chromosomes. And if this figure is abnormal, then it refers to aneuploidy. And non-disjunction, which we've already gone over, the unequal distribution of chromosomes during cell division, can lead to aneuploidy. So this particular karyotype is Down syndrome. And in Down syndrome, on the 21st pair of chromosomes, there is 3 rather than a pair of 2. Now, as I did a little bit more research to expand my own knowledge outside the scope of the curriculum, I was looking at the procedure needed to test if the embryo has aneuploidy. I found out that as women age, so does their genetic integrity of the eggs, and at the age of 35, women in Australia are strongly encouraged to get an invasive procedure called amniocentesis. It was interesting to find out that there are less invasive tests available but are deemed less cost effective, so they are less accessible. Okay, so here students need to use a human karyotype to identify ploidy changes and predict a genetic disorder from given data. So here is one example of a karyotype, and they need to identify the ploidy. the Polluti changes. So from here, as we've gone over, we know that there are supposed to be 23 pairs of chromosomes, but as the student should be able to pick out, on the 23rd pair there is only one. So we can do some different activities with that. Okay, so for this point, I could not think of any specifically inherited mutations off the top of my head, so I did some research and found that blue eyes originated from a mutation to the OCA2 gene, preventing pigmentation of the eye, which I thought was quite interesting. And it is a recessive mutation, meaning that offsprings need both recessive alleles to display this trait. Okay, so looking at the examples that I have here, Mum has both recessive alleles, so she has blue eyes. Dad only has one, so he has brown eyes. And then looking at the offspring, doing a quick cross, which should bring back some Mendel-Punnett squares, PTSD maybe, you'll see that 50% of the offspring have blue eyes. And then looking down, depending on the genotype of the parents, will depend on the variation of the genotype of the offspring doing those crossovers. And that is how the offspring will vary. All right, so now we're looking at this point over here. Now, students don't need to recall specific chemical mutagens, but have an understanding that a large number of chemical mutagens are carcinogenic and interact directly with DNA. So after I did some research, I found an example, and this is outside the scope of the curriculum itself, just to expand my own knowledge, but Agent Orange would be an example of a mutagen. So it was used widely in the Vietnam War, and it caused a lot of birth defects in children whose parents were exposed to the chemical mutagen, and the agent actually contained dioxin. So that's the mutagen in itself, and it's very highly toxic and very carcinogenic. I ran out of room on the slide, so here's the content references that I've used for this presentation. To merge my content with my chosen pedagogy, I have incorporated relevance by using examples of mutations that they may have seen in people of their community. I'm hoping that with a greater education in connecting textbook mutations with real people, that students will see the relevance of learning this content and create a greater air of acceptance. I have divided the content into 3 lessons, given the 3 notional hours. In all the lessons, students will need to learn the basic syllabus as well as answering the overarching relevance question, why are mutations relevant in our community? In the first lesson, I was thinking of engaging them, the first part of the 5E framework, to look at their arms and see if they can find a freckle. Then opening up discussion on sun exposure and how it mutates our skin cells on a DNA level. This will lead into the explore stage of the 5E framework and continue throughout the lesson. In the second lesson, as it relates to non-disjunction and aneuploidy, I was thinking of guiding students to this site or a printout of the site, as there is various disorders on there. And first they will need to answer specific questions of the karyotype, the disorder and the genetic features before turning to how this affects the individual. This will be a part of the explore stage of the framework. They will then need to explain what they have learnt and so on, following the structure. The third lesson, I'm a little bit conflicted and would particularly like some feedback. I would like students to complete an activity regarding an autosomal recessive mutation where they need to predict the variations in the offspring. I've considered the blue eye example as it is widely found in the community, but after more research, I found this mutation is more considered a trait in humans as it has been around for so long rather than a specific mutation, though I'm happy to take on feedback. As a plan B, I've found these other hereditary mutations and wanted to hear what people thought about which ones students might find more relevant: cystic fibrosis, sickle cell anemia, or juvenile glaucoma. Any feedback is welcome. Okay, so now looking at extending my knowledge. To extend my knowledge as a professional, I have turned to the APSTs. I plan to implement the constructive feedback from my colleagues that I'll receive from this presentation. This will build my skills as a teacher and my resilience to take on feedback. To engage in professional learning, I plan to extend my knowledge through ongoing research into the fields of my teaching areas to stay up to date with the fluidity of knowledge. Also note that I have only showed a restricted amount of the research that I have conducted just for the purpose of this presentation. To apply the professional learning, I plan to participate in workshops that are readily available to me to build my skill set and using these skills in the classroom. And thank you everyone for listening, and thank you for your time. I would appreciate any constructive feedback. Just let me know. Thank you very much.